Dear students, you are welcome in this class. I am Dr. Dinesh Kumar, Principal Scientist, Agronomy Division, IERA, New Delhi. And it is 13th of September 2022. I'm really very pleased to be with you this afternoon. And topic is Introduction to Biofertilizer. Dear students, these days we are talking a lot about sustainability or sustainable agriculture. In sustainable agriculture, there are 3-4 issues which are important. First thing is that we need to maintain a good soil health and conserve our resources. This is number one purpose. Second purpose of sustainable agriculture, or we will call it sustainable agriculture, if it is not polluting the environment. It is polluting less to the environment. And thirdly, it is providing a livelihood to the farmers, or farmers are getting sufficient income. So soil health or health of all resources, natural resources, their conservation and better no pollution, environmental safety, and your economics. These are three important aspects which our agriculture should have if it needs to be sustainable. So in this case, as you know, if you want to maintain the soil health, or if you want to improve the soil fertility, then integrated use of nutrients is the best strategy. We have seen results from the long-term fertilizer experiments where it has been suggested that INM is the best strategy. And everywhere people now speak about, let us combine inorganic fertilizer, organic manures, and biofertilizers together to get the best results. And it can preserve or improve our soil health. And there is no pollution if you use biofertilizers. Biofertilizers are part of INM, and they are not pollutant. They will not pollute your environment. And they are the cheapest source, very economic. If a biofertilizer supplies, say, 20 kg nitrogen per hectare, and getting the price of 1 kg nitrogen is about rupees 11. Just assume rupees 11, and 20 kg nitrogen means rupees 220. So 220 rupees have been contributed by a biofertilizer which is costing rupees, say 20-30 rupees. In 20 rupees, you are getting 220 rupees. So that is the really good economics, means it is very economic and it improves the health. Today you will see variety of roles are being played by biofertilizer. They not only solubilize the nutrient and help in their uptake, they also help in producing vitamins, in solubilizing certain nutrients, and also releasing some enzymes. So multiple roles are being played by biofertilizer. So today we will see all these subjects in detail, and overall we will see the definition of biofertilizer and their importance or role and how they are classified, and discuss the role and constraints of different biofertilizers individually. So this may be very interesting lecture, you will realize in the end, and some difficult words, some glossary. Azolla. Azolla is a fresh water fund found in ponds, ditches, canals, paddy fields, fixing nitrogen in symbiotic association with cyanobacteria. So actually these azolla are water funds, and they have different sizes from small to medium size. You can see from 0.5 centimeter to 15 centimeter diameter. You can see azolla, and they are very efficient in photosynthesis, very efficient in photosynthesis. They can double their body weight just in a day. So they are very fast growing under ideal conditions, this azolla. It has got several species, and all the species are doing symbiosis with Anabaena ajoli, that is your cyanobacteria. And azolla many times are undesirable also. In many places, it is being considered as a weed. It can be a weed. And sometimes it causes a lot of problems in the water bodies also, because there it can create some bloom, algal bloom, or shortage of oxygen can lead to death of fish and other aquatic animals. So azolla is not always beneficial. Sometimes it can be harmful also. Biological nitrogen fixation is the conversion of atmospheric nitrogen into ammonia through some biological processes is called as biological nitrogen fixation. And you know it is symbiotic as well as non-symbiotic. Cross inoculation group, a collection of leguminous crop species which are capable of developing nodules when exposed to bacteria obtained from the same group. So we have got cross inoculation groups, means it is a group of crops which are infected by common rhizobium bacteria. Cyanobacteria or blue-green algae. So actually cyanobacteria is a kind of blue-green algae, but all blue-green algae are not cyanobacteria. Okay, it should be clear to you. Cyanobacteria is a blue-green algae, but all blue-green algae are not cyanobacteria. And this cyanobacteria, this particular cyanobacteria, which we are referring to the symbiosis with fern, azolla, is having only chlorophyll A, and it is really not able to do much photosynthesis. It is dependent upon the associated azolla to get the energy, to get the food. And in turn, this cyanobacteria will give nitrogen to the algae, nitrogen to the fern, that is azolla. So cyanobacteria structurally bear a resemblance with bacteria. It resembles like bacteria because it has no nucleus and no organelles. So, and also you can consider it as a kind of prokaryote, and it is not very efficient in photosynthesis. As compared to other bacteria, cyanobacteria have chlorophyll A. Legume, you understand a plant belonging to family Leguminaceae, number of microbes, organism that can only be seen with microscope. This is covered. Symbiosis, see the last one, symbiosis, the living of two dissimilar organisms in close association and cooperation being mutually beneficial. Then AM fungi, I discussed other day also what are AM fungi. I told you ectomycorrhiza and endomycorrhiza. So ectomycorrhiza mostly in temperate areas and on trees, mostly on woody plants. And endomycorrhiza are mostly in tropical, subtropical areas and on our crop plants, our field crops or vegetable crops. So AM fungi is the new name for endomycorrhiza. So it is a type of symbiotic fungi entering the cortical cells of the roots of a vascular plant forming arbuscules. So it actually enters into the cells of cortex cells or parenchyma cells of roots. However, the ectomycorrhiza also enter the plant cells, but it is mostly superficial. It does not go much inside, but AM fungi goes much inside the roots. This is one. And secondly, this AM fungi, you see half or significant growth is inside the plant. However, this ectomycorrhiza, the fungal growth is mostly on the surface of the roots. And then this AM fungi have some structures which are called as arbuscules. Arbuscules, today we will see what are arbuscules. So upon entering the parenchyma, the AM fungus is singular and fungi is plural. Many times students do mistake. They call it fungus or fungi. Fungi is not correct. It is called as fungi and fungus. So in these arbuscules, exchange of phosphorus, carbon, water, and other nutrients take through these structures. So most of the time, this phosphorus is provided to the host, and carbon is taken up by AM fungi. Water is provided to the host, and some other nutrients to the host. but host supplies food to the fungus. Cellulolytic organisms. You know, when decomposition goes, decomposition of organic matter, then cellulose is also there, which is difficult to digest or difficult to decompose. So microbes that break down the cellulose into other compounds are your cellulolytic organisms. Deleterious rhizobacteria. These are non-parasitic. Remember, they are not parasitic. They are not plant bacteria living on or in plant roots. They may live on the plant roots or inside the plant root, but still they are not parasitic and survive on organic compounds released by plant root cells. They inhibit the plant growth by producing some phytotoxic metabolites. So these are called as deleterious rhizobacteria. There are three kinds of rhizobacteria. One is neutral, neither harm nor damage, nor advantage. So neutral kind of rhizobacteria, they will not affect plant at all, positively or negatively. Deleterious rhizobacteria affect the plant negatively. They harm their growth. And plant growth promoting rhizobacteria, they are having positive influence on the plant growth. So PGPR are plant growth promoting rhizobacteria, play a useful role in plant growth other than plant nutrition. They help in the plant nutrition, production of extracellular siderophores, means they can also produce the siderophores, which are produced by plant roots. Antibiosis is also important. Through antibiosis, they can suppress the population of harmful fungi or harmful bacteria in the soil that may attack your crop. So they can save from diseases. They can save the plant from diseases. Production of growth hormones and pathogen suppression. So these are the advantages of PGPR. And PGPR are not different actually. The nitrogen fixing bacteria are PGPR. It is mostly terminology that their benefits other than nitrogen fixation have been counted as plant growth promoting rhizobacteria. So your rhizobium is also a kind of PGPR, azospilum, azotobacter, all are kind of PGPR. I want to tell you here that rhizobium, most of the students know that it infects the leguminous plants. But some people have done some research on rice crop at International Rice Research Institute, and they have published paper. If you treat the rice seed with rhizobium culture, with leguminosaurum, rhizobium leguminosaurum, variety trifolia, whatever it is, rhizobium leguminosaurum has been tried on rice seeds, and it has been found to have PGPR effects, plant growth promoting rhizobacteria has been, this rhizobium has been considered for rice. Just last slide of this glossary. Phytohormones or plant hormones, same. The naturally occurring small organic molecules or substances which influence physiological processes in plant at very low concentration. Rock phosphate, the product obtained from the mining and subsequent metallurgical processing. So rock phosphate, I will speak tomorrow in your practical class. And last item here is your sparingly soluble phosphorus. Sparingly means very, very insoluble, difficult to solubilize. So calcium phosphates such as apatite account for 95% of phosphorus found in primary minerals of the Earth's crust and are commonly referred as sparingly soluble phosphorus. So tomorrow in the practical class, I wish to take two lectures in practical class, one on phosphorus in soil and other phosphorus in fertilizers. So let us come to the main topic, that is biofertilizer. What do we mean by biofertilizer? So we need to know the definition of biofertilizer, because you will be asked sometimes a question, what is biofertilizer? So biofertilizers are the formulations having the living or dormant microbes such as bacteria, fungi, algae, etc. alone or in combination of two or more microbes. So this is not complete definition, but it will work. Biofertilizers are the formulation. You need to remember the word formulation having living or dormant microbes. And you know what are the microbes? Bacteria, fungi, algae, etc. Maybe alone or maybe in combination of two or more microbes. That is biofertilizer. And then you can add that they perform variety of functions, including atmospheric nitrogen fixation, solubilizing or mobilizing nutrients. So there is difference in solubilizing and mobilizing. Solubilizing means the form of the nutrient was fixed form. It was insoluble form. It was not in available form, or you can say the nutrient was in unavailable form. And then to make it available, certain microbes solubilize uh, that unavailable form, maybe organic form, maybe inorganic form, minerals, etc. So that is your solubilization, making it available. Mobilization means it is helping in the movement of ions or molecules or nutrients from one place to the roots. It is helping in the mobilization, like AM fungi, and releasing plant growth promoting substances, that is hormones, for improved growth. Azotobacter is known to produce number of growth hormones. Like others also, some others are also there, which produce growth hormones. Even this Azospirillum brasiliensis also produce number of hormones used to supplement chemical fertilizer and organic manure. So this is how to define biofertilizer. And one more definition for you of biofertilizer. These are the products containing living or dormant microbes, within bracket bacteria, fungi, actinomycetes, algae alone or in combination, which on application help in fixing atmospheric nitrogen or solubilize, mobilize nutrients, in addition to secretion of growth promoting substances for enhancing crop growth. So on the last slide you have seen there were 3 points. All these 3 or 2 points have been merged here. So it becomes complete definition of biofertilizer. Now, role of biofertilizer, how they are important. Number one, fix biological nitrogen in soil, which is readily available to the plant. Whatever nitrogen is fixed by these rhizobium or any biological, particularly rhizobium, that can be immediately used by the plant. Or maybe the nitrogen which is fixed by free living bacteria, that is also readily available to the plants. For example, your rhizobium. So there is concurrent transfer of nitrogen to the plant. As you know that in the fixation of nitrogen, in the beginning, they make very simple amino acids. And simple amino acids are transferred to the legume plant. So that legume plant can convert these amino acids into proteins very easily with less energy. No pollution. As you know, these all kind of biofertilizers, they are pollution free. They will not give any direct pollution. Increase soil fertility and productivity because they are adding nitrogen to the soil and they are also mobilizing phosphorus, solubilizing phosphorus. So therefore, fertility is improved. And as fertility is improved, you can see that productivity is improved. So productivity is the result of improvement in fertility plus the growth promoting action of microbes. They release enzymes, they release amino acids, they release vitamins, they release hormones. All these organic constituents released by microbes or bacteria, they will go into the plant roots. They can directly go into the plant root and improve its growth and productivity. Provide plant nutrient at low cost. I told you that if a bacteria or if N-fixing organism is fixing 20 kg nitrogen, that will be 22 rupees value. And you are investing only 20 rupees. So provide plant nutrient at very low cost. Residual effect in soil. So their effect lasts for many years and need not to apply every year. So once they are applied, they are inoculated in the soil, they survive well and continue for many years, and you will find residual effect of their use. Now, another role, see some more roles of these. Solubilize certain nutrients for crops like mycorrhiza, phosphorus solubilization. Some help in uptake of nutrients, mycorrhiza. Releasing enzymes and vitamins, all these organisms, whether it is Azotobacter, Azospilum, Azomonas, they all release enzymes and vitamins. And what they are doing, enzymes and vitamin, they will improve the defense mechanism of the plant and improve the growth. So defense mechanism here means the insect, the plant will tolerate, or it can become resistant, or there will be minimum attack of insect pests and diseases. Its defense system becomes stronger, tolerating certain biotic and abiotic stresses. So now people have come up that certain bacteria, certain organism, they help to cope up drought conditions. Drought conditions, there are certain bacteria, and they help in uh, uh, tolerating certain biotic and abiotic stresses. So for this course, I think this line is enough, but if you are interested, you can know what are the roles of microbes in improving the defense mechanism, fighting against biotic and abiotic stresses. Now we go to the classification of biofertilizer. Up to now, you have seen some glossary, some definition, what do we mean by biofertilizer? And then you have seen important roles or value of biofertilizers. Now we see classification, how they can be classified, because students can be asked, give us the classification of biofertilizer, of what are different kinds of biofertilizers available. This can be asked in many ways. So they can be classified into 5 groups here: nitrogen fixers, phosphate solubilizers. Phosphate solubilizer, also you can add phosphate mobilizer also. Mobilizer here is not mentioned because that mobilizes other nutrients also. So it is not just phosphorus, they can take up zinc or some other potassium. And then compost accelerator. Compost accelerators are also there, or you can say decomposers. Decomposers are also there. Plant growth promoting rhizobacteria. So this is actually assembly of nitrogen fixers, you can say, nitrogen fixers and others. So others may include your AM fungi here. AM fungi can be included in others. Involved in increasing the availability of soil phosphorus, and recently now some potassium solubilizing and zinc solubilizing biofertilizers are being produced and recommended. However, it has not reached that much popularity among the farmers. Sorry. There may be some problem with the efficiency of K solubilizing and zinc solubilizing microbes. They are not very much in the field, but they are there. At least for the namesake, they are there. So students can remember certain species of organism that solubilize K and zinc. I will give you the list. Now you see the classification, the same is simplified here. You can see biofertilizer and move to the left. First is your nitrogen fixing. So nitrogen fixing can be 2, symbiotic and non-symbiotic. Non-symbiotic, you know the names of non-symbiotic: Azotobacter, Azospilum. And then coming to symbiotic, there can be 2, nodule forming and non-nodule forming. And in this, nodule forming on the roots and on the stem. Can you give any example where symbiotic relationship is there, but there are no nodules? Can anyone give me example that association is symbiotic, but there is no formation of nodule and they are nitrogen fixers?
Sir, azolla.
Exactly. Azolla-Anabina association, symbiotic relationship. Azolla-Anabina.
Nostoc, sir.
Yeah. So that is your non-nodule nitrogen fixers. And stem nodulating? Stem
Sir, Sesbania rostrata.
Good. And roots, all the legumes. In roots, all the legumes are there. Then coming to phosphate solubilizing, they are divided into two: phosphate solubilizers and phosphate mobilizers. And then organic matter decomposer. Because in soil as well as during composting, you need to get your organic matter decomposed. So there are two different kind of groups of biofertilizers: cellulolytic, as the name suggests, that they will break down the cellulose, and lignolytic, they will digest or decompose or oxidize the lignin. So this is how this very simplified classification of biofertilizers. Now we start with nitrogen fixing biofertilizers. Examples are Rhizobium, Azotobacter, Azospilum, Azolla, and blue-green algae. So these are the examples. Nitrogen fixation is done by microbes either symbiotically, that is Rhizobia, or on root nodules, or non-symbiotically by Azotobacter, Azospilum, and Acetobacter, etc. Now we continue 2-3 slide on Rhizobium, and on the right side you can see this nodules are there. Nodules are there on the roots of this is Sesbania, Necha. In Sesbania you have two species. One is Sesbania rostrata, other is Sesbania aculeata. Sesbania aculeata is more frequently used for green manuring purposes, and Sesbania rostrata is normally not used for green manuring purposes because it gets somewhat woody. And many studies were conducted on their nitrogen benefits, and it was found that both are equally effective in nitrogen fixation. So farmers and people mostly use Sesbania aculeata for green manuring. Nitrogen fixation by Rhizobium was first reported by Hellriegel and Wilford 1886, and Beijerinck isolated the bacteria, that was Rhizobium basically, from nodule and proposed the name Bacillus radicicola is the old name of Rhizobium, and the name Rhizobium, Greek meaning rhiza means root, root means living on root, was given by Frank 1889. And Rhizobia are aerobic bacteria. These are aerobic bacteria. Rhizobia is plural and Rhizobium is singular. And one more species, genus has been recorded, that is Bradyrhizobium. The credit is given to, N.C. Brady, whose book you are reading these days. N.C. Bradyrhizobium. So they persist saprophytically. That means Rhizobium can survive without a legume crop in the soil. They can live without legume, they will not die, they will survive. But whenever they get suitable legume, they will infect its roots and become active and start nitrogen fixation. So persist saprophytically means in soil, they can eat the dead matter and they can survive, but they like to grow on legume roots. So that is why many times this inoculation of Rhizobium is not desirable. It is recommended that when you are taking a legume, first time in a new area, then you should get it inoculated with the respective legume. But once it is grown for 1 year, 2 years, then inoculation is not necessary. Not necessary, that means it must be surviving in the soil. So it survives saprophytically in the soil until they infect a root hair. Legume provides Rhizobium with carbohydrate. CHO means here I write In symbols, CHO means carbohydrate. So legume provide rhizobium carbohydrate, means host. Host is supplying carbohydrates and energy to the bacteria, rhizobium, and rhizobium fixes nitrogen for the host plant. So it is give and take. But the enzyme involved in nitrogen fixation is nitrogenase. This nitrogenase is for all, whether it is your blue-green algae, whether it is azotobacter, azospilum, whatsoever is there, the enzyme involved is nitrogenase. So nitrogenase has two distinct proteins, component 1 and component 2. If you see their constitution, component 1 has iron molybdenum protein with 2 molybdenum and 28 iron atoms. And component 2, iron protein of 4 iron atoms. General reaction, you can see this reaction is applicable to other organisms also. N2 plus 6 electrons. These 6 electrons will come from carbohydrate or organic matter supplied by the host. And this is coming from atmosphere. And then ATP is also coming from the host. And then ammonia, 2NH4 is synthesized and ATP converted to ADP and inorganic phosphate. So you can see then this ammonium or ammonia produced is reacting with organic acid and it makes simple amino acids which can be converted to proteins. So this is how this nitrogen fixation happens. I'm not going into much details of rhizobium infection, the growth of rhizobium and so on. That you can read from the book, the different processes in this and how nitrogen is estimated, nitrogen fixation is estimated. What are the methods to estimate nitrogen rate of nitrogen fixation? So you can consult some books if you have interest. So nitrogen fixed by forage legume is higher than the grain legume. So forage legume, can you give me example of forage legume and grain legume? Name of examples or name of the crops or plants which are under forage legume and in grain legume. Students from
forage legume.
Yeah.
Forage legume, barsim.
Barsim, good. And grain legume? Grain legume? You don't know any, really, nobody knows grain legumes. Why do you fear? I think everybody knows. What is grain legume? Example? Okay, good. So grain legume is legume which are giving us grain. So all pulses are grain legumes. Even your groundnut is a grain legume. Of course, it is not pulse, but it is a grain legume. Similarly, groundnut, soybeans, you told soybean is also a grain legume. May not be a pulse, but it is a grain legume. So forage legume may be your barsim and your alfalfa. Alfalfa is your lucerne. So these are. So now question is that nitrogen fixed by forage legume is higher than the grain legume. So what is the reason? I want to know. What is the reason that forage legume like barsim or your alfalfa fix more nitrogen than grain legume? Let's see your green gram, black gram, red gram, yellow gram, whatever is there. So any guess from anybody? So in case of, yeah.
Simply, we don't concern about those plants for their destructive part, that is the grains. That's why they lose less energy for producing those reproductive parts, and those energy are useful for producing nitrogen more amount.
Yeah, very good. So in this case, it is very simple that grain legumes, that most of the nitrogen goes to the grain. But here it can be shared by roots. Means the roots will not be giving much. Much is not required by the plant. and then they can retain more nitrogen, because whatever is fixed, uh, uh, the significant proportion of fixed nitrogen in grain legumes is diverted towards the grain. But here grain is not the purpose. You are not going up to the grain. You are just harvesting the fodder material. So therefore fixation is relatively more in forage legumes than in grain legumes. The residual effect of nitrogen fixed by legumes become available to the succeeding crop, and as measured by crop yields, it is generally equivalent to 40 to 50 kg nitrogen per hectare per season. So on an average, they can leave about 40 to 50 kg nitrogen per hectare per season for the next crop. So this much will remain in soil. Rest will be harvested in the biomass, plant biomass. Now you see nitrogen fixed by different legumes. So in this case, you can see soybean 100 to 150, chickpea 40 to 50, lentil 40 to 68, groundnut 150, field pea 60 to 100, cowpea 90, cluster bean 660 to 150, and clover 100 to 150. See, this is clover. So clover is your maybe berseem or something like that. It is fixing more. And this data is really not very reliable. Anyway, you can check from Brady book. If you want to know more exact amount of nitrogen fixed by different legumes, you please see Brady book. In that book, you will find nitrogen fixation rates. So this data, I have doubt about it. But on an average, if somebody asks you how much nitrogen is fixed by legume, you can say it is 60 to 80 kilograms per hectare. Of course, it will vary with the legume. There are a number of factors that will affect the quantity of nitrogen fixed by a legume. Those factors are important, and we will discuss the factors that will affect the fixation of the nitrogen. And now 1 minute is left. We will go to the other link. Host specificity of rhizobium species, and we call it cross inoculation groups. So cross inoculation groups means one species of rhizobium cannot infect all legumes. A particular rhizobium species is known to infect a specific legume or group of legumes. So cross inoculation groups are simply group of legumes on which the given species of rhizobium nodulase. Means there may be 2 crops, 3 crops, 4 crops. These are cross inoculation, means one's rhizobium can go to other. So cross inoculation groups are simply groups of legumes on which the given species of rhizobium nodulates. So it is group of crops. 1 group, 2 group, 3 group. So in one group, there can be 3 crop, 4 crop. But group 1 crop, the rhizobium of group 1 crop cannot infect the rhizobium of group 2. So to obtain the full benefit of BNF, BNF is biological nitrogen fixation. The correct rhizobia for their legume crop should be used. The right strain should be used, or right species should be used. So I am showing you the list. This is the correct list. Many times students find different things in different books. So this is from Brady book, and it cannot be wrong. So I just cross-checked and verified, and these are the correct groups. Rhizobium, leguminosarum, BV, VCAC, P group, bean group, clover group. So please go through it. Nothing to understand. You have to see them. This is for you to remember few names. I will share this presentation and you can read it. So now nitrogen fixation, as I was discussing, that it is important to know the factors that will affect the nitrogen fixation rate, because you will see if you open 5 different books on this subject, you will find that one crop, all the books are giving different values of nitrogen fixation. So why so much of variation or variability in the data, in the result? Somebody is saying that cowpea can fix 80 kg nitrogen. Some are saying 60 kg, some are saying 120 kg. So such figures are really very, very varying. So nitrogen fixation rate by legume varies with the soil. So if you grow a particular legume in two different kind of soil, or two different kind of soil means their fertility may be different, their pH may be different, so N fixation may be different. Climatic conditions, same legume but climates are different. Somewhere climate may be favorable, somewhere climate may not be favorable. Then crop species. So definitely the amount of nitrogen which is fixed by one species may not be same to the other species under the identical growing conditions. And N fertilizer. How this N fertilizer will affect the N fixation, I will show you later. How the rates of affect this N nitrogen rates or fertilizer rates. So if you are growing first time this legume, then inoculation is must. You must inoculate. If low availability of iron, molybdenum, sulfur, effect of nitrogen fixation means a soil which is deficient in iron, molybdenum, sulfur, because they are very, very necessary for nitrogen fixation. So nitrogen fixation will be low if content of these element is low in soil. Legume rhizobium association function best in soils that are not too acidic, means soil environment reaction, it should be neutral to alkaline. Then the result will be better. And bradyrhizobium, however, can tolerate considerable acidity. It can tolerate considerable acidity, but most other cannot tolerate acidity, and N fixation is less under acidic condition with the comparable situation. Optimum availability of water and other resources. Definitely it is required. Crop management practices like plant species, high level of available nitrogen depresses biological nitrogen fixation. High fertility, it will not fix more nitrogen because under those conditions, there is no need to fix the nitrogen, which is already available to the plant from the soil. So plant will not give much cooperation to the bacteria. So now you can see how this fertilizer affects the nitrogen fixation. So you can see this picture very carefully, and on the x-axis you get increasing amount of nitrogen fertilizer applied. So one legume crop is grown here. Legume crop is grown. On x-axis you have nitrogen application rates and they are increasing, and on y-axis you have amount of nitrogen in legume plant. How much nitrogen is accumulated? Assuming that this is the level of total nitrogen accumulated in the plant. So you can see initially the levels of nitrogen application or rates of nitrogen application are very low. Low supply of nitrogen through fertilizers. Then you can see contribution from biological nitrogen fixation is very large. But if you go on increasing the amount of fertilizer applied to the legume, the contribution of biological nitrogen fixation decreases. And in the end, you can see if you have very high rates of nitrogen fertilizer application, you can see the out of total nitrogen, this much nitrogen is coming from fertilizer. So here contribution of legume is minimum. So it is very simple. If you go on increasing the rate of nitrogen fertilizer beyond a certain point, then the nitrogen fixation by legume reduces and uptake from fertilizer increases. So this is the message. So you need to have the lower level of nitrogen. That is why we apply very lower. You can see at lower level of nitrogen, it is doing well, it is doing best. But when you increase the level, higher level, the contribution of BNF reduces. Is this picture clear to you? If any doubt, then I can read some points for you about this picture. If more nitrogen fertilizer added, the BNF reduces. Inorganic nitrogen replaces BFN. BFN, biologically fixed nitrogen. Biologically fixed nitrogen. So inorganic N replaces BFN, saving plant some energy. Because now plant will not give energy to rhizobium. It is getting directly from the soil. That would have been used by nodule bacteria. Plant growth is little affected by the N fertilizer. Means if you are having good quantity of nitrogen, okay, you are not having BNF, but the plant growth will not be affected if it is getting nitrogen from the soil. Rather, there are reports that some people are talking of nitrogen feeding to the legumes. Let us give full dose of nitrogen to the legume as per requirement, and then we will not allow it to fix the nitrogen so that we get more yield from the legume, because it will not waste its energy to the rhizobia. So, but that is just a fantasy, may not be a truth. Certain legume species fix nitrogen so inefficiently that they need external nitrogen application, like phaseolus bean. Phaseolus bean is your rajma, I think rajma or French bean. It does not have efficient N fixation. Uptake of inorganic nitrogen released by mineralization may or may not be much affected by N fertilizer. Uptake of inorganic nitrogen released by mineralization may or may not be much affected by N fertilizer. You can see in this case, in the small strip in between these two, nitrogen fixed by nodule bacteria and nitrogen fixed by added fertilizer. There is one small strip. So you can see it is almost same. It is not affected. Nitrogen from soil organic matter is not affected either by nitrogen fixed by bacteria or by coming from fertilizer. So you can see that the rate of release of nitrogen from the organic matter is same. It is not affected much. So you have seen the meaning of this picture now. Legume, rhizobium, symbiosis, and soil nitrogen. Legume can significantly increase soil nitrogen and benefit non-fixing species. Some direct nitrogen transfer may take place via mycorrhizal hyphae connecting two plants. So two plants can exchange nitrogen during current season when they are growing in the field. The vigorous development of a grass in a legume grass mixture is evidence of this rapid nitrogen release. So you can find some intercropping systems where legume is there. In one plot, you can have legume as intercrop. In another, you can have the pure crop of the cereal. Then you will find that the growth of legume where you had intercropping, growth of cereal in legume intercropping, you will find better growth of cereal. So you can see this example, dear students, and this is example, I mentioned the reference also. So ryegrass is a cereal plant. Ryegrass is a cereal plant, and in one case, you can see the ryegrass was grown alone, means pure cropping of the cereal crop. And you can see this is mixed cropping of ryegrass with a clover, means cereal plus legume cropping. And in this case, it is ryegrass or cereal crop alone. So you can see on y-axis nitrogen concentration in ryegrass. So ryegrass, when grown alone, it has just 1.8% nitrogen concentration. And when this ryegrass is grown with a ladino clover legume plant, then you can see nitrogen concentration is 3.4%. So why this change in concentration of nitrogen in the ryegrass? Because they are growing simultaneously. That means the nitrogen has been transferred from legume to non-legume partner. So it proves that nitrogen can be transferred concurrently from legume to the associated cereal crop. You can read it. Azotobacter, I think you have idea about it. Non-symbiotic aerobic living organism, free living and fixed atmospheric nitrogen in different crops. It can be inoculated, means the culture, artificial culture can be inoculated with the seed, with the seedling, or it can be directly applied to the soil. Foliar application is also useful. It can be foliarly applied, and carrier-based and liquid-based forms are available. These are the culture made by scientists or made by companies to give it to the farmer for commercial use. Also synthesize some growth hormones. Azotobacter can synthesize gibberellin, cytokinins, and it can fix about 10 to 20 kg nitrogen per hectare per year. The quantity of nitrogen fixed is low, but it is coming in almost in free. But it has some growth promoting influence also. Azospilum is spiral shaped microaerophilic, means it needs little oxygen or partially aerobic gram-negative N-fixing bacteria, and they fix in the association with grass roots. So they live in the vicinity of grass roots and fix nitrogen, isolated by badgering. Badgering are also isolated your rhizobium in 1925 and named Spirillum lipoferum, the first name of Azospilum. And name Azospilum was given by Terran, and it fixes more nitrogen than Azotobacter, 20 to 40 kg N per hectare in association with cereals or grasses. Hormones are indole acetic acid, gibberellic acid, cytokinase. And 3 species are there. One is Azospilum brasiliensis. It was isolated from a Brazilian soil from Brazil. Therefore, name came Azospilum brasiliensis and Azospilum lipoferum. Lipoferum is because fat bearing. And third species is Azospilum pellicle. So 3 species are there. Now coming to Azolla Anabina association, let us have idea on Azolla. These are fresh water found in ponds, ditches, canals, and paddy fields. Fixes nitrogen in symbiotic association with cyanobacterium, that is blue-green algae, Anabina azoli. So Anabina azoli is a one species of blue-green algae. There are 5 species of Azolla. Azolla pinnata, one is there. It is most important, occurs in Southeast Asia and tropical Africa. A. caroliniana, found in North and South America. A. filiculoides, cold tolerant in temperate areas. A. microphylla, heat tolerant, and A. nilotica from Nile in Africa. In India, mostly people use this A. pinnata and A. microphylla are important. And Azolla is requiring a sufficient supply of phosphorus for normal growth. This is very important. For it, nitrogen is supplied by blue-green algae, but Azolla requires your phosphorus from the soil. So what is there? Here I am teaching you about Azolla. That does not mean that this Azolla is not having BGA. This Azolla contains your BGA or your cyanobacteria is there in the cavities of Azolla. It is living symbiotically with it, and it takes carbohydrate from Azolla. So this symbiosis can fix 25 to 40 kg nitrogen per hectare per crop. I have taken this matter from Brady book, Nature and Properties of Soils. So it is also a better food for fish, ducks, and pigs. So it has been also used as a fodder or as a forage material for animals. It is being fed. Used as a green manure crop in China, Vietnam, and some southeastern countries. So it contains 2.08% N, 0.61% P2O5, and 2.05% K2O, and CN ratio is 14.0:1. Maybe 14:1. 14 here means 14:1. 14 carbon, 1 nitrogen. So it can be decomposed very fast. Now let us get idea about blue-green algae. On your right side, you are seeing the picture. These are Azolla. These are Azolla. You cannot see cyanobacteria through your naked eyes. These cyanobacteria are living in the cavity of the these leaves of the plants. These algae or cyanobacteria, you cannot see them through naked eyes. You can see them only by microscope. But what you are seeing is Azolla. So BGA or cyanobacteria, they are filamentous, photosynthetic, aerobic, nitrogen-fixing organisms. Commonly available BGA species are nostoc, Anabina, Olocida, Tolipothrix, and Calothrix. These are different species of blue-green algae, but among them, Anabina azoli is the most important one. And here I want to tell that other species can survive without Azolla, but Anabina azoli cannot survive without Azolla. You should remember it. So it is by default there in the Azolla. All kind of Azolla, this Anabina azoli is there. And this nostoc, Olsoria, and Tolipothrix, they can survive independently also. They do not need this symbiotic association. Symbiotic is for Anabina azole. In paddy fields, they fix about 25 to 40. So this is actually joint fixation. Joint fixation means joint content or uh, 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 because it is joint effort, you can say it is because it is fixing, but it is being held by azolla. So therefore, in paddy fields, they fix about 25 to 40 kg nitrogen per hectare per season. And this is coming from atmosphere. You can see the picture. I got it from Dr. Y.V. Singh, who is also scientist in agronomy. So you can see this. Thank you to Dr. Y.V. Singh for giving this picture. And you can see these are all azolla. Azolla you can see. And their roots are hanging in the water. Their roots are hanging in the water. And you can see this is for cyanobacteria. You cannot see cyanobacteria. This is some material in which they have grown it. So this is a culture medium. Culture medium may be Multani mitti. You must be knowing about Multani mitti or some other things. And now we see about phosphate solubilizers. Just a minute. Phosphate solubilizers and K and zinc solubilizers. Compost accelerator PGPR AM fungi. So phosphate solubilizers, microbes or bacteria, you can see PSM, phosphate solubilizing microorganism. When we see microorganism, that means we are not specifying whether it is fungi, whether it is bacteria or some other, or is it mixture of all. When we say PSM and when we say PSB, that means it is surely a bacteria, phosphate solubilizing bacteria. So there is hardly any difference. You can call it PSB or PSM. So it may include your bacteria, fungi, yeast, and actinomycetes. PSM may include your bacteria, fungi, yeast, and actinomycetes. So these are phosphate solubilizing microbes. Produce, how they solubilize phosphorus. So they produce number of organic acids like citric acid, glutamic acid, lactic acid. You can go through it and solubilize the native or sparingly soluble phosphorus for plant uptake. So these acids produced by these microbes solubilize the inorganically fixed phosphorus. They are not solubilizing or decomposing the organic phosphorus, but they are solubilizing the sparingly soluble P for plant uptake, that is inorganic phosphorus. Recommended with rock phosphate for solubilization of the latter. Because you know in rock phosphate you do not have any water soluble phosphorus. Rock phosphate has 0% water soluble phosphorus and 0% citrate soluble phosphorus. Rock phosphate has insoluble, citrate insoluble phosphorus. So therefore it has no soluble phosphorus. Then you can use certain PSB or PSM to solubilize the rock phosphate and to make the phosphate available. So phosphate solubilizing bacteria are Bacillus megatherium variety phosphaticum, Bacillus polymixa, Bacillus subtilis, Pseudomonas striata, agrobacterium species. So these are species of phosphate solubilizing bacteria. And then phosphate solubilizing fungi, PSF, Aspergillus avamori, Penicillium digitatum, sorry, it is Penicillium bellagi, and yeast Saccharomyces species or Saccharomyces cerevisiae is also there. Actinomycetes includes Streptomyces and Nocardia. So all these organisms can solubilize the phosphorus. And fungi are, of course, better phosphorus solubilizer than bacteria. They may often solubilize 10 times or more phosphorus than bacteria. This is very general kind of a statement that fungi are more efficient in phosphate solubilization than bacteria. Now, K-solubilizing bacteria. Now, they are not in the market, but they are under research, and certain bacterial species solubilize K-bearing minerals and convert K to soluble forms. So inorganic K can be converted into simple available forms or soluble forms. Some positive effect on soil fertility, K uptake, and crop productivity are reported by many scientists. However, a remarkable inconsistency observed means they do not give response always. Sometimes they are effective, sometimes they are not effective. That is your inconsistency in response across crops, regions, and climates. Examples, at least one or two examples you can remember: Acidothiobacillus ferrooxidans, penny bacillus species, Bacillus mucilaginosus, and so on. Similarly, you can see zinc solubilizing bacteria. Fixed zinc can be solubilized. You know how zinc is fixed by Bacillus subtilis and Thiobacillus thiooxidans. So these are there. And then compost accelerators. Some microbes used for making compost and in situ decomposition. In situ means decomposition in the soil of organic residue. There are two groups of compost accelerators. Cellulolytic, that is decomposing cellulose organism. They decompose the cellulose. Trichoderma viridi, Aspergillus niger. So these microbes have multiple roles, multiple uses. This Trichoderma viridi, on one hand, it is used to control some fungal diseases. On the other hand, it is also acting as a cellulolytic organism. Aspergillus niger, Chaetomium abuneus, and you can read them: Clostridium, Nocardia, and Streptomyces. So you get variety of organisms. Some are bacteria, actinomycetes, or fungi, but mostly they are fungi, cellulolytic organisms. And lignolytic, breaking down the lignin and lignified materials. These materials are part of organic matter. So organisms are Clavaria species, and next one is very difficult. I cannot read it. You can see it, and Humicola species. Now we come to the fifth group of biofertilizer, that is PGPR. As I told you, they have multiple roles. For example, azospilum and azotobacter, or even rhizobium. You studied them as nitrogen fixers. So nitrogen fixing is one job. Other job, they can act as a plant growth promoter also. So PGPR by microbiologists have put another group of organisms, they call it plant growth promoting rhizobacteria, PGPR. Or sometimes they call it organism also, plant growth promoting fungi, plant growth promoting organisms, or microbes, PGPM, plant growth promoting microorganism. So it depends what name you want to give. So they reside in or near the plant roots and promote the growth. Three kinds are there. One is beneficial. Beneficial is Azotobacter, Azospilum. Deleterious, I think I have shown you some example already, and there are some neutral groups. So the beneficial free-living soil bacteria are called PGPR, beneficial one. Examples are Bacillus and Pseudomonas, and Rhizobium also you can include. PGPR may affect the plant growth indirectly or directly. So they play a variety of roles. What are roles of PGPR? Beneficial one, fixed nutrients in soil, Azospilum brasiliensis, production of phytohormones, indole acetic acid. You have seen that Azotobacter release indole acetic acid and some release gibberellins, facilitating uptake of certain nutrients like Bacillus subtilis. Bacillus subtilis is known to mobilize or solubilize phosphorus. Bacillus subtilis increases phosphorus solubilization. It is responsible for solubilization of phosphorus and also disease suppression in paddy, in rice, Bacillus subtilis. Lessen or prevent the incidence of diseases like Bacillus subtilis in rice. It is written. Biocontrol agents. Some can be used as biocontrol agent. Now, finally, we come to the last group, which is also important. This is arbuscular mycorrhiza fungi. So up till now, I was little fast because the number of slides is 54. I have covered 41. So now we can be slow bit. You have any question?
No, sir.
Yes. Anybody has any question?
Sir, what is PCM and PSB? What is composition of PSM, sir?
What is the composition of PSB? Phosphate soluble.
PSM, sir. PSB, sir, phosphate solubilizing bacteria, but PSM, sir.
Phosphate solubilizing microorganism. So if we say microorganism means it will include all kind of organism. It will include your actinomycetes, fungi, bacteria, and even yeast. This PSM means phosphate solubilizing microorganism. And if we say phosphate solubilizing bacteria, then only bacteria we need to tell. I have given you the list somewhere here. It is here. So if we call phosphate solubilizing fungi, so you have Aspergillus avamori, Penicillium digitatum, and Penicillium. So list is here. These all are actually phosphate solubilizing microorganism. And if you want to specify, you can specify. If you are talking of only bacteria, then say PSB. If talking only fungi, say PSF. And you can also make new term PSY, phosphate solubilizing yeast. So this is in our hand, how we can write and represent. So is that clear? Is that clear to you?
Yes, sir.
Okay. Now 10 minutes are left and I can finish in next 10 minutes. So now we go to arbuscular mycorrhiza fungi. It is symbiotic association between fungus and roots of vascular plants. Fungus colonizes host plant, that is intracellularly. Fungus will enter into the cell and your ectomycorrhiza are intercellular. If they enter the plant cortex, they go into intercellular spaces, not in the intracellular. So these endomycorrhiza are intracellular and they are called as AMF, arbuscular mycorrhiza fungi, because they make arbuscules in the plant cells, therefore they are AMF. Before they were known as VAM, 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 vesicular arbuscular mycorrhiza, V for vesicle, means they were making vesicles. But it was found that all the endomycorrhiza were not making the vesicles. All fungi were not making the vesicle. Therefore, name was changed to AMF. And you can see the ectomycorrhiza, I've already told you, are associated with roots of only 10% plant families, and rest 90% plant families are colonized by this AM fungi. Ectomycorrhiza are primarily consisting of a hyphal sheath or mantle. Mantle means thick growth outside the root and a heartic net of hypha. Now you see about AM fungi. In AM fungi, aseptate hypha. I think you know there is no division or septa that divide a hyphae. In AM fungi, aseptate hypha enter root cortical cells and form arbuscules. So first it will enter, the hypha will enter, and then it will make arbuscule. On the right side, if you can see the picture, it is not very clear, but it is in the cell making the arbuscules. So arbuscules are intracellular. They are in the cell, tree-like branching. You can see the branching here. A structure in root cortical cells. Arbuscules are essential location for exchange of carbon compounds from plant. So here the plant will donate or plant will give carbon compounds of food to the plant, to the fungi, and mineral nutrients will be given by this arbuscule to the cell, plant cell, particularly phosphorus. Plants provide sugar to the fungi. So this is kind of symbiotic association. And you can see in this picture very clearly, I think. And here, this hypha is passing through intercellular spaces, but it is entering in the cell, into the cell. And this structure, if you can see, is your arbuscule. Therefore, name arbuscular. And rest are plant parts. You can see root here. And sometimes it may make your reproductive chlamydospore also. You need not to bother about other parts of the fungi. Just see that it is making arbuscule in the in the cells, and it is the site, it is the place for exchange of nutrients from fungi to plant and plant to fungi. You can see with more clarity this picture. So arbuscule is inside the cell. You can see it is inside the cell. This is the fungal structure in soil, this fungi structure. You can see these pictures later. Now, why they are important AM fungi? They increase the root volume and absorptive surface of roots. 10 to 1,000 times more root surface increase because they are very fine. Their surface area is more. Total surface area is more. Increase uptake of soil resources, nutrients and water. They acquire phosphorus, highly mobile in soil. Acquire phosphorus, highly immobile. So therefore, phosphorus uptake is increased. Extension of the plant root system, acquiring phosphorus from the areas which are not accessible by the plant roots. So they can go to the places where roots cannot go. Release tightly bound soil nutrients, phosphorus, iron, and others. Means they solubilize the nutrient. Protection of host from abiotic and biotic stresses. So mycorrhiza is important in protecting the host from abiotic and biotic stresses. You know about biotic stresses, attack by organisms, other organism to the plant, or abiotic, your temperature, water, these are abiotic stresses. The external hyphae improves soil aggregation. This is also improved. They are improving physical property of the soil. Reduce erosion and maintain soil porosity. Important role. They bind particles together. They must be releasing some material into the soil that bind the particles. Conduit for communication between linked plant. So two plants can exchange their contents or their nutrients with each other, and particularly nitrogen. This is the way you can see the nutrient can be transferred between two plants. And suppose this is legume plant and this is non-legume plant. So nitrogen can be transferred to non-legume plant. And this is published in Nature magazine. So it cannot be wrong. You can check it. So the bridge is made by mycorrhiza and through which the exchange takes place. Suppose this plant is having more sugar. So it can give sugar to it and take nutrient from it. So this is how this friendship is going. Not obligatory, but they are helping each other. In the last figure, what is happening, you can read it. You can read the same thing what I told you is written in these two pages, but please read it. It is very important how the exchange is happening. Here also, in this picture also, it is shown that how nitrogen is transferred from legume to non-legume when they are growing together. Direct nitrogen transfer by mycelial network. So here this mycorrhiza is helping in the exchange of nitrogen from legume to non-legume, and you can read it. I have given the complete reference here. So most mycorrhizal species exhibit low host specificity, allowing for connection between dissimilar plants. Even DC oilseed can be connected to the legume through the this mycorrhiza. A lot of studies has been conducted on this aspect. You can see white clover can transfer nitrogen to ryegrass. Persimmon clover can transfer to maize. P2 barley. There are 1,000 papers which are supporting that mycorrhiza can transfer nitrogen from legume to other materials from non-legume to legume. In the end, you can see some biofertilizer constraint. Upkeep and maintenance requires special care. Storage is difficult. They cannot be stored. They are to be stored at right temperature, right humidity. So storage is the issue. Otherwise, their cells will die at high temperature or at very low temperature. So you need to keep them at right temperature for their viability. And they partially meet the nutrient requirement. So one cannot wholly dependent upon biofertilizers alone, but they can supplement. They can supplement to other sources, organic manures and chemical fertilizers. Highly acidic or alkaline soils are not fit for biofertilizers. And then heavy metals should not reach the field. Otherwise, they can reduce the growth of these biofertilizers. So thank you very much. It completes the biofertilizer